Okay, hello. My name is Jarl Gunnar Flaten. I'm a researcher at Sintef Nord in the group on computer science and machine learning. And I'm here to present our work on automated identification of barbed wire jellyfish in citizen science images. This is joint work with my colleague Erling Devold and Tone Falkenhaug, who is a senior researcher in the plankton department of the Institute of Marine Research. And this activity is carried out through the JellySafe project, which is funded by the FHF. Now, I want to start with an interactive exercise, a show of hands. So please raise your hand if you know what a barbed wire jellyfish is. Okay, that's not everyone, so good thing I brought an image of one. And now, keep your hand up if you've seen one in real life. Okay, so that's a few. And finally, keep your hand up if you've submitted a photo to the citizen science service, Dugnad for Havet. So that's just me. So I encourage you, if you see one, then you can go to Dugnad for Havet and you can easily submit one, a picture of one, or any other interesting marine life that you may see. And there's an app in development that will make this a bit smoother. This is a citizen science portal where anyone can submit images, and then there are experts at the IMR who will validate the species that you see. And this data set is used for various purposes, including biodiversity monitoring and understanding species distribution. And in this activity, we've used these images to train detection models for this specific species of barbed wire jellyfish, also called apolemia. I'll be using the two names interchangeably. Okay, so I want to tell you a bit about why we're doing this, how we're doing it, and the downstream applications of this work. So, as you may know, there are various challenges associated with barbed wire jellyfish that the aquaculture industry has faced in the recent years. So this is a stinging jellyfish that organizes into long string-like colonies. Here's an image of one. And when it comes into turbulent water or into contact with something like fish pen nets, then it fragments into clouds of these stinging cells, and that's what's causing issues in large quantities. So here's another image where you see fragmentation throughout the whole image. And this organism is distributed in the Pacific, Atlantic, and Mediterranean seas. And in recent years, we've seen it along the Norwegian coast. Mainly in the fall, between September and December, were the concentrated observations in 2023 and 2024. But the last winter of 2025, we've also seen some observations sporadically. So it remains to see if we'll see them the end of this year as well. As I said, large invasions have caused issues, so disruption of aquaculture operations and mortality events, causing economic losses. And the JellySafe project is assembling both knowledge on the biology of this organism as well as the spread in order to produce mitigation measures and operational measures in order to prevent harm to the industry. And our detection activity is part of this. So let me go through the three levels of ambition that you can have when you try to detect something in an image. And these are the steps that we've gone through. So we started out with whole image classification, and we've got good results on that. And then we went on to do something a bit more specific, which is so-called bounding box detection or object detection. Now this turns out to be a bit unsuited for the shape or morphology of this organism. So we went a step further to what we call instance segmentation. This is still ongoing work in progress, in particular with respect to swarms and quantification. So I'll go through in more detail for each of these. Okay, so starting out with whole image classification. This is the simplest task where you say for an image, does it contain the organism you're interested in? In our case, it's apolemia. And, or is it marine background, so the negative class? And it's a yes-no question for each image. And you see here a grid of 16 images that our model has classified. And when we've trained this on the Dugnad Verhaven image data set, we achieved a cross-validation accuracy of 98%, which is satisfactory, we think. So the benefits of this approach is that it's quick to train, we achieve high accuracy, and it's easy to benchmark. So we tested out different models and we benchmark against the state of the art on other marine data sets. And it's easy to compare the results. It's a yes-no question of whether an image contains this organism or not. But the cons, there are some drawbacks to this approach. And one of the drawbacks is that it's very prone to bias. And this is especially challenging when you have a sort of unsystematic data set that is like the ones you get from citizen science. So you could imagine that maybe all the apolemia images also contained like fish pen structures or something correlated in the images, but which has no actual relation to the thing you're interested in. And that would be a bias that we wouldn't be able to discard in these predictions. And we wouldn't see it because we don't have any insight into what exactly is the model looking at in the image in order to produce its prediction. So it's less transparent than the other methods we'll be looking at shortly. In addition, it does not produce any level of quantification. It's a yes-no for each image. You can't say how many. So therefore, to address these drawbacks, we went on to do so-called bounding box detection. So this is a detection method which produces bounding boxes around each object that it detects. So we have two images here. In the leftmost image, there are two contained like intact colonies of apolemia that the model detects. And then it's a bit more of a chaotic environment on the rightmost image, but the model correctly identifies all the different ones, even given the fish nets all around it. It's also a method that supports multi-class detection, so you can have multiple objects in a picture, and they can have different species or classes. And expanding to other species than apolemia is an ongoing discussion that we have with the IMR that requires also sufficient data to carry that out. So these are some pros of this approach. We also get a better quantity estimate. You can count the boxes, but a box isn't really a good quantity estimate. Maybe if we consider these two boxes here, if you think about the strings within these boxes, they seem maybe approximately as long. If you were to unwind it, this is a bit more tangled than that one, even though the boxes have different sizes. So it's a better quantity estimate, but still not a perfect one. Another benefit here, though, is that the predictions are more transparent. We can easily see if the model is misidentifying a piece of rope or something that might look like apolemia as apolemia, and we can adjust accordingly. And we actually had to do that. So, like I said, this is a better approach than the previous one on full image classification, or more specific benefits from it. But in order to have some level of quantification that's more suited to this morphology, we decided to go a step further to instance segmentation. So this is where we have pixel-wise class predictions. So we see a mask here on the right. And I'm demonstrating that we have an image here that we're running our algorithm on. We don't just have to do it on still images. So here we get, like I said, pixel-wise predictions. And the prediction is as detailed as it can be. And we're working on improving our masks for swarms and especially chaotic images, like one of the ones we saw earlier. Now, a drawback of this approach is that it's very laborious, both in terms of manual work, if you have to annotate, and in terms of GPU power, RAM, and then time spent training these models. So In my final time, remaining time, I want to talk about the downstream applications of this work. And there are 3 in the JellySafe project. The first one is to automate the validation of images that are submitted to the Dugnad for Havet citizen science portal. So if you can submit a photo of currently apolemia, I mean this is work in progress, and have instant feedback on whether the image contains the species, then this is this instant feedback is good for learning for the user, but it's also something that will save the expert validator time. If we have quicker validation results on these submissions, we can tie into and trigger an early warning and risk forecast, which is also future work in this project. And we can. The idea is to run a drift model to identify downstream localities that might want to know about incoming invasion of apolemia. And for this, we require better swarm detection and quantification, like I mentioned. And another application is just quantity estimation as a means on itself, which builds on top of segmentation. And I'm sure there are many other applications that you can think of. So either of this specific work or of transferring these methods to other problems and domains. And I'm very eager to hear about ideas. We have our own ideas as well. So don't hesitate to contact any of us. We're all ears. So thanks for listening. And I think there's some time for questions, maybe.
Thank you. Any questions from the audience? I don't see any. Okay, I have maybe a small one.
Okay.
So you say you could maybe do some prediction then to warn maybe the farmers on incoming jellyfish. Do you have any idea what they could do then to prevent that from actually reaching inside the cage? Or
we have ideas, yeah. So this is part of the project activity. I don't think I want to come out with a list on a standing right here now.
Okay, no problem. Thank you so much.